Welcome student to my channel Bio Tutor. How are you? Today I have come some important part, that is C3 cycle or dark phase of photosynthesis or light independent phase of photosynthesis. Okay, and here the dark phase is also known as Calvin cycle or C3 cycle. Why? Because here first of all one stable compound will be producing, that is known as phosphoglyceric acid, where the number of carbon will be 3. That's why it is known as C3 cycle. Fixation of carbon dioxide in phase will be happening in different steps. Not only the C3 cycle, C4 cycle, and another is Cassoulian acid metabolism process. Clear? The C3 cycle, the first stable compound is PGA, that means phosphoglyceric acid. Okay, and where one substrate is needed, that is RUBP or ribulose 1,5-bisphosphate. Okay, another one in C4 cycle, here also one compound will be producing, that is oxaloacetic acid, where the number of carbon will be 4. That's why it is known as C4 cycle. Clear? And another process I told you, CAM, that means Cassoulian acid metabolism process. This kind of photosynthesis will be happening at night in the absence of sunlight. And some kinds of plants, as for example I am giving Cassouliaceae, the plants which are under the family of Cassouliaceae, we can see this process. Clear? And here the process, when I am giving one example of one plant, that is one Opuntia plant, one cactus plant, we can see this process. Here the photosynthesis will be happening during night. But today's topic is C3 cycle. It will be discussed later. Okay? So, in case of Calvin cycle, here not only the Calvin and his workers, Bassam, scientist Bassam and Benson, they will doing this work. They have done this work. Okay? And this Calvin cycle will be happening by through some different steps. First one, fixation of carbon dioxide, that is known as carboxylation process. Second one is reduction process. From phosphoglyceric acid, phosphoglyceraldehyde will be producing, and here this process is known as regeneration process, where NADH2 will be acting as reducing agent by giving out is hydrogen. Okay. Next one, the process, there are another two processes. One is synthesis of glucose and another one is resynthesis or regeneration of RUBP. What is the full form of RUBP? RUBP full form is ribulose 1,5-biphosphate. And here I'm telling one thing, here the RUBP is needed as substrate. Okay, and carbon dioxide is also needed as raw materials by which is absorbed, which is taken by green plants to produce one one product, that is the first stable product is known as phosphoglyceric acid. But here one enzyme is needed as biocatalyst, that is known as Rubisco. Here you just see that CO, the last word CO is very important. This Rubisco enzyme will be performing dual functions. While it will be acting as while it will be reacting as biocatalyst in the fixation of carbon dioxide in Calvin cycle, at that time it is known as RUBP carboxylase for the reaction, for catalyzing the reaction of fixation of carbon dioxide in C3 cycle. Clear? But sometimes due to high temperature and due to presence of huge oxygen, Calvin cycle will not be happening At that time, this Rubisco, this enzyme will be catalyting, will be acting as RUBP oxygenase, and this effect is known as Warburg's effect. Okay, at that time glucose will not be producing. Okay, at that time again the carbon dioxide and water will be forming. So here it will be happening only due to presence of high temperature, high sunlight, while the intensity of light will be increasing, and due to presence of huge oxygen in air. And at that time it will be acting as oxygenase enzyme. So the enzyme is Rubisco. Clear? But C4 cycle will not be happening. In case of C4 cycle, here one special anatomy, special structure is present. It will be discussed later. That is known as Cranch anatomy. And for this Cranch anatomy, this may not happen. This will not be happening. Here the temperature will not be increasing and this Warburg's effects will not be happening. But in C3 cycle, while the temperature will be increasing, and at that time this process, that means Warburg's effects will be happening, and at that time glucose will be breaking down into carbon dioxide and water again, and at that time this enzyme will be acting as RUBP oxygenase. Clear? So this is the process of Calvin cycle, and here two things are needed which are produced as assimilatory power in the light phase. And what are they? ATP and NADH2. And this ATP and NADH2 will be needed here for completing this cycle. Clear? So now I am showing you how is it happening through one cycle. Okay? This is the picture of Calvin cycle. According to the name of scientist Calvin, this cycle is known as Calvin cycle, or dark phase or light independent phase of photosynthesis. Okay, it occurs during day, but here sunlight is not needed. And in light phases, two products will be produced as assimilatory power. What are they? First one is ATP and second one is NADH2. Okay, and here first one substrate is needed here, that is RUBP, ribulose 1,5 biphosphate, and where the number of carbon will be 5. Number of carbon here will be 5. Clear? So 6 molecules of RUBP where the number of carbon is 5 will be reacted with carbon dioxide. Plants, green plants, take this carbon dioxide from air as raw materials for fixation. So here the fixation of carbon dioxide will be happening. 6 molecules of carbon dioxide will be reacted with this substrate, that is known as RUBP, or ribulose 1,5 bisphosphate. And one enzyme is needed here, that is Rubisco. Rubisco means RUBP carboxylase oxygenase. Here it will be reacted, it will be acting as carboxylase enzyme for the fixation of carbon dioxide. Okay, so this Rubisco will be needed here as biocatalyst or enzyme to form the first stable compound, that is phosphoglyceric acid. Okay, 12 molecules of phosphoglyceric acid will be producing here. Clear? And it is the first stable compound of this cycle. That's why, and where the number of carbon is 3. That's why it is known as C3 cycle. I told you first, and this step where the fixation of carbon dioxide will be happening, only this step is known as carboxylation process. Okay, this step is carboxylation. Next step, from these 12 molecules of phosphoglyceric acid, it will be reducing to produce 12 molecules of phosphoglyceraldehyde. We know that while the acid will be reducing, it will be producing some aldehyde substance. So here 12 molecules of phosphoglyceric acid will be producing phosphoglyceraldehyde through some products. First of all, it will be producing 12 molecules of 1,3-diphosphoglyceric acid. Here the number of phosphate is 1, but here I have written 1 in one position. There one phosphate is present, and in 3 position another phosphate is present. So here number of phosphate is 2, because here the ATP which is producing which was produced as assimilatory power in the light phase will be added here. Okay, and from here phosphate will be added here. That's why one phosphate is here, another phosphate is coming from this ATP to form 1,3-diphosphoglyceric acid, where also the same thing, but the number of carbon will be 3 here. So RUBP here number of carbon 5, here the PGA where the number of carbon is 3, and in diphosphoglyceric acid here the same thing also, here the number of carbon is 3. Okay, so the process from where the PGA will be forming PGLD through producing 1,3-diphosphoglyceraldehyde. This process is known as reduction process, oxidation reduction process, with the help of one dehydrogenase enzyme. Okay, and here this NADH2 will be oxidized into NAD again. Okay, after giving the hydrogen it will be oxidized. So at that time it is acting as reducing agent. While it is giving the carbon hydrogen, at that time it will be acting as reducing agent. Okay, but you just see here 1,3-diphosphoglyceric acid. From here 12 molecules of phosphoglyceraldehyde produced. That means one phosphate again will be coming out, will be given out as inorganic phosphate. So how many molecules here? 12 molecules. Because here 2 molecules of PI will be coming out, will be given out in each step, and if this cycle will be happening for 6 times, so the number of inorganic phosphate will be 12. Clear? Each step 2 molecules of inorganic phosphate will be given out, but it will be completing through 6 cycles, 6 times. So here the number 2 into 6 means 12 molecules of inorganic phosphates will be coming out. So here 12 molecules of PGLD, that means phosphoglyceraldehyde, will be producing, and where the number of carbon also the same thing, 3. Here one thing. So now the next step, 12 molecules of phosphoglyceraldehyde. Among these 12 molecules of phosphoglyceraldehyde, 10 molecules of phosphoglyceraldehyde will be again regenerating, will be again resynthesized RUBP. Again will be resynthesizing RUBP through some intermediate, through forming some intermediate compounds. Where the number of carbon will be 3, 4, 5, 6, 7 like that. Okay? Tetros, erythros, tetros, pentos, hexos, pseudoheptulose like that. Through some intermediate compound, 10 molecules of PGLD will be produced. Again the RUBP where the number of carbon will be 5. And 2 molecules of phosphoglyceraldehyde will be forming glucose. So this step where the 2 molecules of phosphoglyceraldehyde will be producing glucose, this step is known as synthesis of glucose. So first one, fixation of carbon dioxide, that means carboxylation process. Second one, reduction process from phosphoglyceric acid to phosphoglyceraldehyde. And phosphoglyceraldehyde, among these 12 molecules of phosphoglyceraldehyde, 10 molecules again will be forming the RUBP, that means regeneration of RUBP or resynthesis of RUBP, and 2 molecules of phosphoglyceraldehyde will be forming glucose, synthesis of glucose. This process is also known as the synthesis of glucose from phosphoglyceraldehyde, is also known as glycolytic reversal process. Why? Because it is just reversible process of glycolysis. Why? Because in glycolysis, which will be occurring in cytosol, there glucose will be, through different process, will be producing pyruvic acid. Okay, but I am not coming to that process, to the pyruvate. But here one compound will be producing, that means phosphoglyceraldehyde from dihydroxyacetone phosphate with the help of one enzyme, that is triose phosphate isomerase. Here the just opposite, just reversible from this here process from here. From here the phosphoglyceraldehyde will be producing dihydroxyacetone phosphate, and dihydroxyacetone phosphate will be producing glucose through some different, with the help of some enzyme and through some processes, again glucose to phosphoglyceraldehyde, and here phosphoglyceraldehyde to glucose. So just reversible process of glycolysis. Okay, that's why it is known as glycolytic reversal process. And this process will be first explained, the synthesis of glucose from two molecules of phosphoglyceraldehyde will be first explained by was first explained by scientist Calvin. It was first explained, synthesis of glucose from two molecules of phosphoglyceraldehyde was explained by scientist Calvin. That's why the synthesis of glucose is known as Blackman's reaction. Again I am telling, the synthesis of phosphoglyceraldehyde, synthesis of glucose from phosphoglyceraldehyde was first explained by one scientist, that is Blackman, not Calvin. So this reaction, synthesis of glucose from phosphoglyceraldehyde, this process is known as, according to the name of the scientist Blackman, this is known as Blackman reaction. But scientist Calvin and his co-workers observed they observed and they said that regeneration of RUBP was happening from 10 molecules of PGLD through some intermediate compound. So the regeneration of RUBP from PGLD is known as Calvin cycle. But synthesis of glucose from phosphoglyceraldehyde is known as Blackman reaction, because Blackman explained that and Calvin explained that PGLD will be regenerating RUBP again. Okay, so this is the process of Calvin cycle, and formation of glucose is known as Blackman reaction. Clear? So this is the Calvin cycle or C3 cycle. Another cycle will be happening next time, that is C4 cycle. Clear? Thank you, students.